That's the wrong size. No, restart it, no! Welcome to the MD Toxin Lab at the University of Maryland Eastern Shore. And so what we do is we put them on pipette tips, and you can pull out specific amounts that you want. So this one only goes up to 10 microliters. How many?
1,000.
Perfect. Vortex. We use a vortex whenever we have samples in, and we want to make sure that there's fully a uniformity between the sample inside that vial. So you just press it on. You can put your finger on, and it'll start shaking like this. Now start shaking up the small little vial. Now, is it grams of force? It's grams of force, right? We take off this lid, and there are a total of about 24 little small vials that we can put in here. We have to make sure that the vials are adequate enough to undergo the grams of force that the centrifuge puts on it. So there are specific types of vials that we put in here solely for centrifuge. But then once we put all our samples in, we do this, we close it, we set our grams of force, we set a temperature, and then we set however long that we want this to run for.
Soil, like DNA extraction, like they taught us how to do it real quick, and that's all I know so far.
So this is our very big fridge. It is set at negative 40 degrees, and this is where we keep a lot of our samples, so that makes sure that their samples don't go bad and that they don't start heating and start giving us bad results in the end. So this is where we keep everything really important, everything needs to be frozen. This is our UV machine, so whenever we're done with the gel electrophoresis, we put our Agros solution gel onto this. We turn it on. I'm not going to turn it on right now because it's very powerful, very dangerous right now. But we put it on here, and then we can see clear banding with the UV light.
Hi, my name is Jalen. I'm a senior general agriculture major with concentration in pre-vet. And right now we're in the insectary, and this is where we keep all our mosquitoes. We're doing the Culex pipiens species, and we're focusing on PFAS and the two compounds, PFBA and PFOA. So we have them in different numbers of the concentration. So we have it from 10 all the way to 5,000. And then we also have the control here. And then right now in these containers are just the larvae and maybe some of the pupae. And then eventually once we see pupae, we'll take them and then put them into their cages. And I can check out the cages too. Distilled water, and then we put the pupae into here, and then they hatch into the mosquito, and you can kind of see them flying around. And once we see the mosquitoes, I have a little tube that I can show y'all, and I have to take the mosquito and suck it through the tube, and then put it into a different tube to then put it into the -80 freezer. I won't be able to demonstrate this because we try to do the adults all at one time, so if I bring the adults out, and I know we're about to go to the greenhouse about it, but I can explain what this is. I take it into the cage, and I just try to suck up all the mosquitoes. And there's a filter on here so that the mosquitoes don't go into my mouth. And after that, I take another tube and blow the mosquitoes out into the tube. In my notebook, I just have it in different sections, the PFOA, PFBA, and then the control. And then when I come here, I just count the larvae at the top and then count the larvae at the bottom. If there's any dead, I take the dead out, and then we count all the exoskeletons in here. And then there's adults, we do plus, and then for pupae, we also add it, and it's all cubes. This is the -80 freezer, and this is where I keep all the adult mosquitoes in. Once I transfer it into the tubes, and I can just show you. Here's all the mosquitoes there. Culex pipiens.
Oh, okay.
Hello, my name is Tayden Kelly. I'm a senior and my major is biology, biological sciences, bachelor's of science. And this is the envitoxin lab. And so here my role is to basically grow these plants with different parts of PPT, so parts per trillion of a different concentration of PFBA. Now PFBA is perfluorobutanoic acid, which has many effects on the development of the fall armyworm. And so here we're looking at the real world practice of what concentration in soil and through the irrigation of PFBA, how does it affect the larvae, and do they grow faster? What internal changes are they making? And I guess overall, how will this impact the future at a real world scale? So this is where we make our different parts per trillion. So in this one, we have 100 parts per trillion of the PFBA. So we just added 100 microliters into a 20-liter container here. As for our 500 parts per trillion, we add 500 parts per trillion inside this little 3-liter bottle right here. And then for the 0 parts per trillion, we just add normal DI water.
Again, my name is Jalen, and my focus is on PFAS, more importantly talking about PFBA and PFOA and how it will affect Culex pipiens within mosquito species.
Again, my name is Tayden Kelly, and I work with PFBA and the developmental effects of the fall armyworm using PFBA.